Chapter 14, Section 6, Oxygenic Photosynthesis. Okay, the oxygenic photosynthesis, photosystem 1, photosystem 2, 2 becomes 4, 1. All of this you've seen, you've talked about before. But, and there's the little asterisk, the disclaimer, the players here are going to be slightly different. Everything you've learned about in other courses, chances are it's going to be based on plant biology. Here it's not going to be plant biology. Here we're going to be focusing on the different prokaryotes and how they do it. So we're going to see that photosystem 1 is going to be an iron-sulfur type complex. Photosystem 2, Q-type, the quinone. PS1, PS2, do that Ziem scheme that you've seen before. 2 transfers to 1 in the linear, or 1 transfers back to 2, 2 to 1, 1 back to 2 for the cyclic that you've seen and talked about before. Differences here, eukaryotes, it's in chloroplast. Here in the cyanobacteria, it's stacked membranes that we've talked about before, and these membranes are just offshoots, or inshoots, I should say, of the cytoplasmic membrane. And it's everything you've talked about before, you've seen before. So here it is all spelled out, but I'm going to do it here from this figure. Refer back to the previous slide in your handouts. Photons of energy hit chlorophylls that are acting as the light harvesting complex. Okay, they pass on and excited to P680, the special chlorophyll, which is basically the 2 chlorophylls as one. Passes it on up to the Q proteins. Q proteins passes on that excited electron to an electron transport chain that's going to bleed off the energies, pump hydrogens. Pass it on over here to another set that are going to prepare it and pass it on to photosystem 1. Photosystem 1, which is going to do the same thing again. Pass on the excited electron to P700. P700 kicks it up, pushes it back, and then now does this cyclic thing. Except, every so often, the FNR protein is going to reduce NADP to NADPH, and so that way we don't end with oxygen anywhere here acting as the final electron acceptor. Here the final electron acceptor is NADPH, which is then going to go use it in the light independent reactions. Over here, photosystem 2, to replace the electron that P680 gave up, water. Water will be split. Oxygenic. Boom. Anoxygenic can work roughly the same way. So you can utilize an anoxygenic photosystem in oxygenic phototrophs. What happens here? PS2 is blocked. Okay, photosystem 2 gets blocked. Okay. If this occurs, photosystem 1 can operate on its own. It's just going to have to look for, its excited electrons from different places that it's then going to cycle it back, electron transport, cycle it back, electron transport, and so on. So that way there's still going to be some NADPH and some ATP made so that you can still have carbon fixation, the light independent reactions occurring somehow. Cyanobacteria and a few others can utilize hydrogen sulfide, if necessary, to replace the splitting of water for photosystem 2. So that, yes, while photosystem 1 is trying to go, these can shunt, or they just completely have lost the ability to utilize water as the electron, or as the electron replacer. So they use hydrogen sulfide, but they're still going to be using oxygen at the end of their oxidative phosphorylation steps. that are occurring separate of this. So they are aerobic. They are doing photosynthesis, but they can be using hydrogen sulfide in place of water to replace their electrons. Or they could be facultative anaerobes that just choose not to split water. All sorts of different ways, all sorts of different permutations for this. What this is showing here are bubbles, but the bubbles are not gas. They are sulfur. The splitting of hydrogen sulfide by this bacteria, Oscillatory Laminecta. So it's going and splitting hydrogen sulfide, and here is the byproduct, which in plants that would be oxygen bubbles. Here it is sulfur bubbles, S2.